Hello friends, welcome to Dr. Varsha Gaitonde's channel. In this video, we shall see the most important information about Xenia and Metaxenia. As you know, Xenia and Metaxenia are always the confusing phenomena which are observed in some of the plant species. So, let us see the theoretical portion, then I will explain you how exactly it happens. The definition of xenia is it is a effect in plants, that is the effect of pollen on seed and fruit of the fertilized plant. The effect is separate from the contribution of pollen towards next generation. Term was coined in 1881 first time by Fouquet to refer the effect of maternal tissue including the seed coat and pericarp, but at the time of endosperm was also thought to be a maternal tissue and the term became closely associated with endosperm effect. Xenia and Metaxenia are the phenomena dealing with the effect of pollen from different sources on certain characteristics exhibited by seeds and fruits in a variety of species. That means Xenia is the effect of pollen on the seed characters and Metaxenia on the fruit characters. Please kindly remember these two things. One of the most familiar example of xenia is the different color that can be produced in maize by association of alleles by individual pollen grains or assortment of alleles by individual pollen grains. The endosperm tissue is not produced by the mother plant but is produced or product of the fertilization and genetic factors carried by the pollen which affect its color. Example, yellow seeded variety may have its yellow color determined by a recessive allele. If it receives pollen from a purple seeded variety, then it has one copy of the dominant allele for purple color and one copy of recessive allele for yellow seed, and resulting cob will have the same yellow and some purple seeds. Such maize cobs are cultivated for decorative purposes. In the Xenia effect, I'll just draw here diagrammatically what will happen. I already told you, Xenia effect is always observed in maize plant. Okay, here I am just drawing the plant. Here you can see the cob or the silk portion, and at the top you will get the tassel portion. As the definition says, xenia is the effect of pollen on the seed characters based on the dosage effect, dominance of one character over the other, and many more things. Here, what will happen in maize? As you know, the position of male and female flowers are entirely different, and this is a monoecious plant. Okay, and when the pollens are taken from here, or when the pollen falls from here to here, it is a self fertilization, and if the pollen falls from the adjacent line, then it is a cross fertilization, or we may say it as production of hybrids. If it is from the sister plant, then it is called as sibbing. Okay, so you should always remember three things. One is selfing, when it is falling from the same source, then from sister plant it is a sibbing, and when it is from the other variety or other inbred line, then it is called as crossing or cross fertilization. So when we observe the xenia effect, we observe it when we get the pollen from other kind, or it is of different inbred line. Okay, from the same source, or if we go for selfing, the effect of xenia won't be observed, because when we are selfing, the same genetic expression will be there, and but when we are crossing from the different source, the pollen effect will be clearly observed. Two things should be remembered here. Xenia is observed when there is cross between different inbreds or different varieties. Okay, and the second thing is xenia is expressed in the same genetic or same generation. That means if we are crossing two plants, okay, when the female produces a cob, at the same time only we will observe the Xenia effect, not in the next generation. If we are observing it, the variation in the F1, what we will say, we will talk about the different phenomena like heterosis and all. But here, because of the masking effect of the alleles and the dominance of one trait over the other, we will observe the Xenia effect. That is one thing. And always the effect is observed in endosperm, not in the embryo. This also you need to remember. Again, we will come back to the theoretical portion. When in course of hybridization, pollen changes the appearance of the hybrid seed still on the mother plant. The phenomena is known as Xenia. This is apparent in the case of endosperm character. Formerly, it was thought to be a direct effect of the male on the female exerted through the pollen. While the zygote is formed by the union of male and female gametes so that it is diploid, and with two sets of gametes, the endosperm mother cell in angiosperms is formed by the fusion of three nuclei, first two embryo sac nuclei and then a male gamete. So the endosperm mother cell is triploid, having three sets of genome. As you might have seen the female part during flowering, or the gamete of the female, how it will be having 8 nuclei condition. Okay, so whenever you will find the endosperm, it is always the three set of genomes, or we may say the endosperm will then be three genes of the allelomorphic pair, which may be symbolically represented as a capital A, small a, like that. In different symbols it can be represented. It is found that in most cases where strict dominance is observed, if only one of these three genes is a dominant gene, then the endosperm shows dominant character in spite of the simultaneous presence of two recessive genes. That means here male and female united, endosperm mother cell is formed by the fusion of three nuclei. This much is clear to you. Then first two embryo sac nuclei. Okay, then the male gametes, the fusion of egg as well as these two tissues. Okay, means these three will be forming one set, that is in 3n condition. Okay, in three cases, if only one is showing dominant character, means the entire endosperm will show the dominant character irrespective of the number of recessive genes. In Xenia, the endosperm characters are evident one generation earlier. That means the characters are not expressed in F1, means the character which need to be observed in case of F1 are observed even in the similar generation where you have crossed. In ordinary hybridization, the hybrid seeds and ordinary self-fertilized seeds on the mother plant look like. The hybrid character becomes apparent only when the hybrid seed grows in the F1 plant. On the other hand, the hybrid seed become once distinct from the self-fertilized seed because of their endosperm character. This is because the endosperm develops fully while is still located on the mother plant, that is on the plant representing the previous generation. इसको मैंने previous slide पे explain किया है. This is just theoretical portion I have given like this because if you want to note it down, you can just write it down in your notes. The phenomena can be clearly seen when a maize plant with white grains is crossed with one with purple grains. ये कोई ज़रूरी नहीं है कि पर्टिकुलर मतलब कलर के पोलन सोर्स ही लेना पड़ेगा, बट if dark colored means you can clearly distinguish the effect. Okay, if some of the flowers in female inflorescence of a pure breeding white grained maize plant are intentionally or accidentally pollinated with pollens from a pure breeding purple grained maize plant, the mutant maize cob will show the hybrid grain as purple. मतलब यहाँ पे एक experiment किया गया है, and this is a suggestion to you also. If you want to see the Xenia effect clearly, what you should do, you should plant 2 lines. Okay, better you plant 3 lines. Okay, one line will be the lines, as you know, the line will be having homozygous kind of individuals, so that what you should do, in some plants you do selfing, and in some plants you just make the crosses with the adjacent inbred line which is purple. Okay, here for your better understanding I'll keep it as P and this as white W. Okay, so when the purple charactered pollen is placed on the silk of white silk, then what will happen, the purple character will be expressed on the cob in the same generation when the cobs appear. Thus, this purple F1 character will be seen on the mother parent itself as an expression of Xenia. Similarly, the F1 plant, when fully grown and self-fertilized, will show the F2 segregation of endosperm color on the cobs that will still be on the F1 plants. The proportion of purple to white grain on such cob will be 3:1. Here, the segregation ratio is shown in F1 instead of segregating in case of F2 when Xenia effect is observed. Similar endosperm characters are common, especially in the different cereal crops, which are mainly endosperm crops. Starchy dominant glutinous recessive grains of rice, as also the starchy as dominant and sugary as recessive grains of maize, are similar pairs of endosperm characters which show Xenia. That means Xenia effect is not only limited to maize, but it is also observed in rice and other major cereal crops. This is how the characters are observed in maize, diagrammatically shown here. First, here you can see the female parent is having very simple kind, white or yellow kind of cobs. Okay, and male is having purple color or dark colored grain. And when you cross these two, not in F1 but in the same generation, when the cobs are produced, those cobs will be having the effect of pollen also. Okay, here you can see in this picture clearly different coloration of the cobs. This is purely with the effect of Xenia if it is expressed in the same generation. Coming to the effect of Metaxenia. As I have told you, Xenia was coined by Fouquet, and he did many experiments to understand that phenomena. After that, he worked with Metaxenia also, but the term Metaxenia was coined by Swingle in 1928. This is sometimes used to describe the effect on purely maternal tissue such as seed coat and pericarp. That means there is always confusing definition between Xenia and Metaxenia, but nowadays the scientists made it clear to distinguish between Xenia and Metaxenia. Metaxenia is a condition during hybridization where during hybridization the alleles of one locus behave as a double dose for the other and make it as a recessive allele. Okay, that means one dominant allele is present, it acts as a double dose, means duplication effect, and masks the effect of recessive allele. Masking effect by this double dose of one allele is called metaxenia. That means single allele will express in double dose and it will suppress the recessive character, that is nothing but metaxenia. Condition is found in aneuploids where segregation is prevented. This you should always remember. Metaxenia is common in case of aneuploids where segregation is prevented. The effect of the pollen on the maternal tissue enclosing the seed. This effect is especially visible in the development of apples. Metaxenia is the effect of pollen grain on the testa and fruit valve. Example, date palm. Here I have quoted two examples. One is apple and one is date palm. Okay. Many times you might have seen, if this is an apple, you might have seen the variegated color like this, some patches or some bar like structures on apple, which makes the apple very special or look very attractive as comparing to the pool of other apples. That is nothing but the effect of metaxenia, and this effect is mainly seen in fruit walls and testa. Testa is the outer covering of seed and fruit wall as the outer covering. We'll come to the final part. Here one important question is there: Xenia and Metaxenia terms are related with. Four options are given: pollen culture, only endosperm, xylem and phloem, pollen and endosperm. So you should always remember that this is an effect and related to pollen and endosperm. So far we have seen that Xenia and Metaxenia effect, as Xenia effect is mainly seen in maize, the popular example. Not only in maize, other crops like in cereals, rice and other crops are also prone to Xenia effect. And when we talk about Metaxenia, it is the effect of pollen grain on testa or fruit wall. That means double dose of a character in dominant form will mask the effect of recessive gene, and this is commonly observed in apple and date palm. Xenia means direct effect of pollen grains on the seeds and fruits outside the embryo. That means the effect is not on embryo but always targeting the endosperm. By this information, I hope that you might have understood the clear difference between Xenia and Metaxenia and the effects of these particular phenomena on different crops, and the effect, the major thing or the major beauty of these effects are the variegation or the attractiveness in color formation, as well as the physical or the phenotypic appearance of particular crop. Okay, thank you. I hope the information furnished by me will be informative to you, and if you like this video, please do not forget to like, keep on sharing, subscribing and commenting. Thank you once again.